Welcome back to another Sleepy Steve story. Before we drift off tonight, remember to subscribe to help Steve-O out, and I will keep making more sleepy stories to help you study gently, revise quietly, and settle your mind at the end of the day. Before we drift off tonight, let us set our coordinates for biology. We are moving into Unit 3 now, and Unit 3 begins by looking much more closely at the cell. Not just the cell as a whole, and not just the organelles that sit quietly inside it, but the molecular instructions that help living things grow, maintain themselves, repair themselves, and pass information from one generation to the next. Tonight, we are going to begin with nucleic acids. That sounds like a heavy phrase, but it is really just the name for a group of molecules that store and use biological information. The two nucleic acids you need to know are DNA and RNA. DNA is the molecule that stores genetic instructions. RNA is the molecule that helps use those instructions, especially when cells make proteins. So, as you settle in, imagine we are looking at a quiet cell under a very gentle microscope. We are not rushing. We are simply moving closer and closer, past the cell membrane, past the cytoplasm, towards the nucleus, where the chromosomes are folded away. And inside those chromosomes is DNA, long, delicate, organized, coded DNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. Both are nucleic acids, and both are made from smaller building blocks called nucleotides. A nucleotide has three parts. There is a phosphate group. There is a pentose sugar, which means a 5-carbon sugar. And there is a nitrogenous base. Those three parts together form one nucleotide. In DNA, the sugar is deoxyribose. In RNA, the sugar is ribose. That is one of the key differences between the two molecules. You do not need to make that more complicated than it is. DNA contains deoxyribose. RNA contains ribose. The bases are also important. In DNA, the 4 bases are adenine, thymine, cytosine, and guanine. We often shorten them to A, T, C, and G. In RNA, the bases are adenine, uracil, cytosine, and guanine. RNA does not use thymine. RNA uses uracil instead. So the first calm comparison is this. DNA has A, T, C, and G. RNA has A, U, C, and G. Now, DNA is usually described as double-stranded. 2 strands run alongside each other and twist into the famous double helix. RNA is usually single-stranded, although it can fold back on itself in certain situations. For VCE biology, the basic comparison is clear. DNA is usually double-stranded. RNA is usually single-stranded. The 2 strands of DNA are held together by complementary base pairing. Adenine pairs with thymine, cytosine pairs with guanine, A with T, C with G. This is one of the most important patterns in biology. It comes up again and again. It matters in DNA structure. It matters in DNA replication. It matters when cells use DNA to make RNA. And it matters when students are asked to explain how genetic information can be copied accurately. So let us say it again, gently. In DNA, adenine pairs with thymine. Cytosine pairs with guanine. In RNA, because uracil replaces thymine, adenine pairs with uracil when RNA is being made from a DNA template. Cytosine still pairs with guanine. This is the quiet logic of the molecule. The bases do not pair randomly. They pair in a specific way. That specificity allows information to be copied and used. Now let us think about the structure of DNA a little more carefully. The outside of the DNA molecule is often called the sugar-phosphate backbone. That backbone is formed by alternating sugar and phosphate groups. The nitrogenous bases point inward towards the center of the molecule, where they pair with bases on the opposite strand. You might imagine a twisted ladder. The sides of the ladder are the sugar-phosphate backbones. The rungs of the ladder are the paired bases, adenine with thymine, cytosine with guanine. The ladder twists, and that gives DNA its double helix shape. This structure is not just decorative. In biology, structure and function are always connected. DNA is suited to storing information because the sequence of bases can carry a code. DNA is suited to being copied because each strand can act as a template for a new complementary strand. DNA is suited to being stable because it is double-stranded and because the sugar-phosphate backbone helps protect the bases. If an exam question asks why complementary base pairing is important, you can say that it allows DNA to be accurately replicated because each base on the original strand determines the base that will be added to the new strand. Now, let us move from DNA as a molecule to DNA as information. A gene is a section of DNA that codes for a functional product. In many cases, that product is a polypeptide, which can become part of a protein. Some genes code for functional RNA molecules. Either way, a gene is not the whole DNA molecule. It is a particular sequence within the DNA that has a biological role. A genome is the complete set of genetic material in an organism. So a gene is a section of DNA. A genome is the whole collection of genetic information. This distinction matters. A human has many genes, but the human genome is the whole set of DNA. A bacterium has genes too, and it also has a genome, although its genome is arranged differently. A virus may have DNA or RNA as its genetic material depending on the virus. In eukaryotic cells, such as plant and animal cells, most DNA is found in the nucleus. It is organized into chromosomes. Eukaryotic cells also contain some DNA in mitochondria, and plant cells contain some DNA in chloroplasts as well. In prokaryotic cells, such as bacteria, the DNA is not inside a nucleus. It is usually found as a circular chromosome in the cytosol, and bacteria may also contain small circular DNA molecules called plasmids. For VCE biology, you want to be comfortable moving between these levels. Nucleotides form DNA. DNA contains genes. Genes are located on chromosomes. Chromosomes are part of the genome. Nucleotide, DNA, gene, chromosome, genome. Each level is connected, but each word has its own meaning. Now let us consider coding and non-coding DNA. Not all DNA directly codes for proteins. Coding regions of DNA contain instructions that can be used to produce polypeptides. Non-coding DNA does not directly code for polypeptides, but it can still be important. Some non-coding sequences help regulate gene expression. Some are involved in chromosome structure. Some are transcribed into RNA molecules that have functions of their own. A common mistake is to think that non-coding means useless. That is not a safe assumption. Non-coding DNA may not code directly for a protein, but it can still play important roles in controlling when, where, and how genes are expressed. This matters because Unit 3 biology is not only about having DNA, it is about using DNA. Cells contain the same genetic instructions in many body cells, but different cells use different genes. A skin cell and a nerve cell can contain the same genome, but they do not express all the same genes. Gene expression helps explain how cells can become specialized. We will return to gene expression in the next episode. For now, simply remember that a gene must be used or expressed for its information to have an effect on the cell. Now let us move into DNA replication. DNA replication is the process by which DNA is copied. It occurs before a cell divides, so that each new cell can receive a complete copy of the genetic information. DNA replication is described as semi-conservative. That word is important. Semi-conservative replication means that each new DNA molecule contains one original strand and one newly synthesized strand. Imagine the original double-stranded DNA molecule unzipping. The two original strands separate. Each original strand acts as a template. New complementary nucleotides are added to each template strand. The result is two DNA molecules. Each one has one old strand and one new strand. That is semi-conservative replication. One original strand is conserved in each new molecule. The accuracy of DNA replication depends on complementary base pairing. If the template strand has adenine, the new strand receives thymine. If the template has thymine, the new strand receives adenine. If the template has cytosine, the new strand receives guanine. If the template has guanine, the new strand receives cytosine. This is why the base pairing rules matter so much. They are not just something to memorize. They explain how DNA can be copied. Several enzymes are involved in DNA replication. You do not need to turn the process into a frantic list, but you do need to understand the basic sequence. The DNA double helix unwinds and unzips. The hydrogen bonds between base pairs are broken. Each strand is used as a template. Free nucleotides are matched to exposed bases. The sugar phosphate backbone of the new strand is joined together. The two new DNA molecules form. DNA helicase is commonly described as the enzyme that unwinds and separates the DNA strands. DNA polymerase is the enzyme that adds complementary nucleotides and helps form the new strand. DNA polymerase also has a proofreading role, helping reduce errors during replication. So, if you need a simple exam sentence, you might say, DNA helicase separates the two strands by breaking hydrogen bonds between complementary base pairs, and DNA polymerase adds complementary nucleotides to each template strand to produce two identical DNA molecules. That is a strong sentence. It links enzyme action to molecular structure and to the outcome of replication. Now, replication must be accurate, but it is not perfect. Errors can occur. An error in the DNA sequence is called a mutation. Some mutations have no effect. Some may alter the protein produced. Some may be harmful, and some may be beneficial, depending on the context. We will explore mutations in more detail later, but it is useful to see the link now. DNA is copied so that information can be passed on, and when that copying changes the sequence, variation can arise. This is one of the great connecting ideas in biology. DNA stores information. DNA is copied. DNA can change. Those changes can affect proteins. Proteins can affect traits. Traits can affect survival and reproduction. And over generations, inherited variation can contribute to evolution. You can feel how Unit 3 and Unit 4 connect already. Now let us compare DNA and RNA more fully. DNA is usually double-stranded. RNA is usually single-stranded. DNA contains deoxyribose sugar. RNA contains ribose sugar. DNA uses thymine. RNA uses uracil. DNA is generally more stable and acts as the long-term store of genetic information. RNA is generally less stable and acts in the use of that information, especially during protein synthesis. There are different types of RNA. Messenger RNA, or mRNA, carries a copy of genetic information from DNA to the ribosome. Transfer RNA, or tRNA, helps bring amino acids into position during translation. Ribosomal RNA, or rRNA, forms part of ribosomes. You do not need to master all of protein synthesis tonight, but you should know that RNA is not just a weaker version of DNA. It has its own roles. DNA is like the stored instruction library. RNA is like the working copy and the molecular helper that allows the instructions to be used. Let us pause and gather the key language. A nucleotide is made of a phosphate group, a pentose sugar, and a nitrogenous base. DNA is a double-stranded nucleic acid that stores genetic information. RNA is usually single-stranded and helps use genetic information. A gene is a sequence of DNA that codes for a functional product. A genome is the complete set of genetic material in an organism. Complementary base pairing means A pairs with T in DNA and C pairs with G. In RNA, A pairs with U. DNA replication is semi-conservative because each new DNA molecule has one original strand and one new strand. These are not just definitions, they are tools. They help you explain processes. If a question asks how DNA structure allows replication, you can talk about the double-stranded structure, the separation of strands, each strand acting as a template, and complementary base pairing, allowing new strands to be assembled. If a question asks the difference between DNA and RNA, you can compare sugar, bases, number of strands, and function. If a question asks why genes matter, you can explain that genes are sequences of DNA that contain instructions for functional products, and that these products can influence cell function and organism traits. If a question asks about the genome, you can explain that the genome is the complete genetic material of an organism, including genes and non-coding DNA. Now, let us imagine the cell again. The nucleus is quiet. The DNA is not floating as a simple loose string. It is organized. It is wrapped around proteins, folded and arranged into chromosomes. The information it carries is not being used all at once. Some regions may be active, some may be silent. Some genes may be expressed in one type of cell and not another. The same DNA can support many different cell types because gene expression is regulated. This is a powerful idea for Unit 3. Cells maintain life by controlling chemical processes and information flow. DNA is not simply a recipe book sitting on a shelf. It is a regulated information system. Different instructions are accessed at different times, in different cells, and under different conditions. But before cells can use DNA, they need DNA to be stable and copyable. The structure of DNA makes this possible. The sugar phosphate backbone gives strength and direction. The base sequence stores information. Complementary base pairing allows copying. The double-stranded structure provides a template. Enzymes manage the process. In Biology Answers, it is often useful to write in cause and effect form. Because DNA has complementary base pairing, each separated strand can act as a template. Because each original strand acts as a template, two identical DNA molecules can be produced. Because replication is semi-conservative, each new molecule contains one original strand and one newly synthesized strand. Because DNA replication occurs before cell division, daughter cells can receive the genetic information needed to function. This is the rhythm of a strong response. Use the biological term, then explain its significance. Now, for a final quiet revision, let us walk through one DNA replication example. Suppose one strand of DNA has the sequence A, T, C, G, G, A. The complementary strand would be T, A, G, C, C, T. A pairs with T. T pairs with A. C pairs with G. G pairs with C. G pairs with C. A pairs with T. If RNA were made from a DNA template, adenine in the RNA would pair with thymine on the DNA, but uracil in the RNA would pair with adenine on the DNA. So when RNA is involved, remember that uracil replaces thymine. This is simple, but it is important. Many students lose marks because they mix up thymine and uracil. DNA uses thymine. RNA uses uracil. Now, let us gently close the episode. Tonight we have moved from the living cell down to its molecular instructions. We have looked at nucleotides, DNA, and RNA. We have seen how genes are sections of DNA and how the genome is the complete set of genetic material. We have explored base pairing, the double helix, and the sugar phosphate backbone, and we have introduced DNA replication as a semi-conservative process that allows genetic information to be copied before cell division. In the next biology episode, we will follow the information further. We will move from DNA to RNA to protein. We will look at transcription, translation, codons, amino acids, and gene expression. But for now, let the main ideas settle. DNA stores information. RNA helps use information. Nucleotides build nucleic acids. Base pairing allows copying. Genes code for functional products. Genomes contain the full set of genetic material, and semi-conservative replication allows cells to pass on the instructions of life. That is enough for tonight. Let the helix unwind gently in your mind. Let the bases pair quietly, A with T, C with G, and let the cell become still. Sleep well.